our today's speaker, Anacs Bушнита, who is now assistant professor at the University of Texas at El-Paso. Anacs Bушниta is, well, on one hand, a young researcher, on the other hand, confirmed researcher. So we can say young confirmed researcher in the field of mathematical modeling and numerical simulations, computing, basically in biological and biomedical applications. And Anas, during his career at the moment is not very long, but already worked on many different topics and very successfully. Since with some people in the Roden University, we discussed mathematical modeling of blood flows and applications. I thought that it might be interesting to ask Anas to present this topic, because Anas is a very high-level specialist in this topic, and well, basically in computer modeling of blood flows with different types of models, not only continuous models, but today we are interested in continuous models. So Anas will present us, I don't know, probably some background, but also recent advances in continuous models of blood flows and their applications. Please, Anas, you can start your presentation.
Thank you very much, Vitaly, for this introduction and for inviting me to give this talk. And thank you, Anastasia, for helping me with the technical preparation for technical details for this seminar. And thank you all for joining me to attend this talk. So for this presentation, it will not be about some original research, but maybe it will be about like a crash course on continuous modeling of blood flow, or like introductory lecture. But of course, I will give some examples from the research that I'm doing, in particular with Professor Vitaly Volpert and other researchers. So this will be the outline of my talk. First of all, I will talk about some physiological and mechanical background of the topic, in particular what is blood and what is the circulatory system. And then I will talk about properties of mechanical and biomechanical properties of blood, what we call rheology, and how we can capture these complex properties using what we call constitutive models of blood. After that, I will introduce two types of models. The first one are single-phase models, and the second are multi-phase models. And in the discussion, I will conclude by analyzing the difference between these different modeling techniques and the different challenges that we have to overcome. So let me start by talking about blood. So I think we all know what is blood. So the first interesting thing about it is that it represents about 10% of the human body weight. So this means that if someone weighs around 70 kilogram, then probably 7 kilogram is, or their body has about 7 kilogram. And for a typical adult body, it has around 5 liters of blood. So probably this corresponds to someone who weighs around 60 kilo or something, because the density of blood is almost 1 kilogram per millimeter, or a little bit higher than 1 kilogram per millimeter. So the second interesting thing is that blood consists of mainly three components. First is the blood plasma, which represents, I think, 50%, around 50% of the blood, and then red blood cells, which represents on average 45%, and the rest is the white blood cells. And here, let me introduce something very important, which is that for these numbers, they are not fixed for everyone. So they can change from one person to another. For example, this 45% of red blood cells, it is what we call the hematocrit, and it can be different from one person to another. So this is like the average value. And then we have this figure here, which give us some numbers regarding how many red blood cells we have, how many platelets, and how many white blood cells, which constitute the three most represented cells transported by blood. So we can say that blood is like a liquid with suspended solids or deformable solids. And then we have this blood plasma, and blood plasma is basically consists mainly of water. So more than 90% is water, and the remaining consists of different proteins and vitamins, hormones, electrolytes, mineral salts, etc. So this is the part of the blood that's make some functions possible, like transporting oxygen, nutrients, and nourishing the different organs and so on. So this is what blood looks like in the microscope. So this is white blood cell, this is like platelets, and these are red blood cells that have this biconcave shape. So also we have this circulatory system, which consists of the heart, the blood vessels, and blood itself. So this is the canals or the vessels that transport blood to different organs of the organism and bring it back to the heart. So basically, we can classify the blood vessels here into two types. So first we have the veins and the venules, and second we have the arteries and the arterioles. And the difference is that for the arteries and the arterioles, for example, the aorta and the carotid artery and so on. So these are the vessels that transport blood from the heart to the rest of the organs. And for the vessels, it's the opposite, which means that the vessels transport blood from organs back to the heart. And naturally, there should be also some areas where the vessels or where the arteries bring blood and then pump it into veins. And these are what we call the capillary beds. So these are areas where that can be found in different parts of the organism, such as the stomach, the lungs, the kidneys, the liver, and so on. And they look like this. So basically, this is a vein, and this is an artery, and we have these different small capillaries, which are blood vessels that take blood from arteries to veins. So there are some distinct characteristics that arterioles have and arteries have that make them different from the veins. So first of all, they have more rigid walls. So their endothelium is more rigid, and also they have high pressure drop, which means that flow in arteries circulates faster than in veins. Okay, with this brief introduction, let me start with describing some mechanical properties of blood. So first of all, as I said at the beginning, the density of blood is approximately a little bit higher than 1 kilo per, I think this should be meter. Okay, sorry for this, meter cube. And this is slightly higher than water. So water is 1 kilo per meter cube. And then the viscosity of blood is also higher than the viscosity of water. So the saying of blood is thicker than water is true in this sense. And the viscosity, so we have the dynamic viscosity and kinematic viscosity. So the dynamic viscosity is about 3 to 4, 10 to the power of -3 Pascal second. And it is different because or due to the, first of all, the hematocrit, how many red blood cells there are in blood, and also the temperature. So as temperature changes, viscosity also changes, and we are going to see this with more details. But in particular, also viscosity grows with the hematocrit. So to introduce, so here we can see, for example, this how viscosity increases with the hematocrit and how it changes with the shear rate. So I'm going to define all of this, but first we need to define one concept, which is the shear stress, and it corresponds to the force that is exerted by fluid on a certain area. So we have the wall shear stress, and it's given by this formula. And we have another concept, which is the shear rate, and it corresponds to the change in two different two layers of fluid, whether it's blood or something else. So with this classification, we have three types of fluids depending on how viscosity change with the shear rate. So if viscosity does not change with the shear rate, then we call it a Newtonian fluid. So this corresponds to this case. And if viscosity decreases as shear rate grows, then we call it a shear thinning fluid. And this is the case of blood. So for blood, if the shear rate increases, and we are going to explain why, then viscosity also decreases. And there is a third case where the viscosity increases or grows when the shear rates increase, and this is what we call a shear thickening fluid. So after that we have defined the shear stress on the wall. So we need to define the shear stress at the bulk of the fluid. And to do this, we can take a look at this picture. So here we have an element of a fluid that undergoes some deformation, and after some small time step, like small time, and we can describe this basically this deformation that was so there are two variables that can be used to describe this deformation, which are these two angles. So these two angles are the difference between this line and this line, and the other angle is the difference between the vertical components before and after this deformation. And so the strain is the difference in this angle minus the difference in this angle. And here we introduce the deformation tensor, which is given like this. So basically, it's the derivation of this. And when we derive this, we know that for very small angles, then they can be approximated by tangents. And so we can obtain these simplifications. And after that, we have our shear stress for the bulk of the fluid. So here it's very important to note that viscosity is not constant, as I have explained before. So viscosity depends on the shear rate, and this is what we are going to explain next. So when we try to measure the viscosity of blood, then we notice that there are three distinct behaviors. So first of all, when shear rate is very low, like below 1 second to the power of -1, then the blood or the red blood cells, they come together and they form these stacks or these layers that we call rouleau. So this looks like this. So it's this red curve, and as you can see, the red blood cells, they are stacked one on the top of the other, and after they make this rouleau, so they make the deformation of the fluid harder, which increase the viscosity naturally. And when we consider moderate shear rates that are under 400 second to the power of -1, then we have two distinct dynamics that can be observed, which are tank treating and tumbling. So tank treating is when the red blood cells move across the fluid, and we can see that its membrane rotates. So the rotation of the membrane, and tumbling is the rotation of the red blood cell itself. And in this case, the blood cells still influence the blood mechanics of the blood, because they can form layers and they can deform in different ways. However, when we increase the shear rate to high values that are over 400 per second, then the red blood cells they lose their resistance, and they become deformed. So they align with the flow, and so the viscosity of the whole blood also decelerates. So now the question is to find some models that can describe this curve that we can see here in the red color. And this is why we introduce the constitutive models. So the constitutive models, they are used to describe the relation between the viscosity and the shear rate. And for example, for Newtonian flow, we don't need constitutive models because the viscosity is constant. So in this case, it's sufficient to take viscosity constant. We don't need to update viscosity for each shear rate value. And as I have said, I have talked about these three different cases of shear thinning and shear thickening and Newtonian flows, and they can be described, or the simplest way to fit this curve is to use what we call a power law fluid model. So a power law fluid model is given like this. So it's K shear rate to the power of n-1, and depending on the value of this parameter n, we can have either, so if n is equal to 1, then this will be equal to k, and we will have Newtonian rheology. But if n is lower or strictly less than 1, then the rheology that we obtain is shear thinning, which means that the dynamic viscosity will be decreasing as the shear rates increase. And of course, if n is strictly higher than 1, then we obtain shear thickening fluid. So this is the simplest model that can be used to fit the rheology of most fluids. However, for the case of blood, this is not very accurate, because first of all, if we look at the asymptotic behavior, so the behavior of rheology near zero and near when shear rates goes to infinity, then when shear rates goes to infinity, the viscosity does not go to zero. So basically, this function, of course, goes to zero if n goes to infinity. And when the shear rate is near zero, then the value that we observe here does not match what happened because of this Rulu stacking. Then we have some sort of stagnation. So the viscosity does not keep increasing exponentially, and so we need to make some changes to fix this model, if we can say it like this. And we introduce two types of viscosities. So we introduce the viscosity at the infinity and at zero. So these are asymptotic viscosities. And then we can describe the viscosity, or the model of viscosity. We can basically scale them using this formula. And here we can have different models, like constitutive models, that give us this very good fit with the data. So we have this Powell-Erin model, we have the cross model, we have the modified cross. So basically, I took this from this work by Professor Adelia Sequeira, and you can see that these are the parameters that fit the data very well. And basically, we need to choose this constitutive model such that they verify two things. So first of all, when the shear rate goes to 0, they need to go to 1. And when the shear rate goes to infinity, they need to go to 0. And so we can find these different models, and all of them, they can approximate blood with very good accuracy. So basically, for me, I use either the Scaro or the cross models, but this is my own. So this is just because I have been using them, so there is no particular reason. But one important detail is that these asymptotic viscosities, so the viscosity at the infinity and at 0, they can change depending on the temperature and the hematocrit. And this is what we can see here, for example. So this is the viscosity that was measured in the lab using mechanic and testing different properties of the blood. And we can see that this blood are given for different values of the hematocrit. So the curves to the low or to the lowest values are for lowest hematocrit. And as we increase the hematocrit, we get away from, so we go a little bit to the top. And we can notice, first thing we can notice is that for very low or for relatively low hematocrits, like for 30%, we can use Newtonian rheology. So here we can see that viscosity does not change too much with shear rate. So the effect of red blood cells is negligible at this point. But as we increase viscosity, as we increase the hematocrit, then the effects of red blood cells become very important. And in some cases like this, it's very important to use non-Newtonian rheology models. So by fitting this data, we can find the asymptotic curves or the asymptotic viscosities that give us these different curves. And we can find this relation that are given as polynomial, that is a function of the hematocrit. So we can use these correlations to find these asymptotic viscosities depending on the different hematocrit levels. So there are some other models like viscoelastic models and that use Maxwell equation and so on, but I think this is like the main, the summary of the rheology of blood flow. And so next I will introduce what we call the single phase blood flow models, and they come from applying the conservation of mass and conservation of the momentum laws. So first of all, blood is an incompressible fluid, and this means that the density is constant everywhere. And so if we use the conservation of mass, which is given by this equation, then we can get rid of this term because this density does not change. And since here the rho is a constant, then we obtain this equation. And this equation is called the continuity equation. And mainly it's used to adjust the pressure. Because when we make calculations with what we call Navier-Stokes equations, then we need to guess and we need to correct the pressure when blood flow changes, since the pressure also influences the movements of blood flow. And this is the equation that we use to correct this pressure. And next we apply the conservation of the momentum law. So basically we apply the same law to the momentum. And here we obtain this equation, but here we have a source term, which means that which correspond to the effects of the shear stress and the pressure and so on. So we can make some development. So we can develop this product, and then we can use the continuity equation so this equation, and after that we obtain this simple formula. And so what remains is to determine the source term. So for the source term, it's given by this this form. So it's minus the gradient of the pressure plus the gradient times the shear stress, and the shear stress is given by this. So by like this, so it depends on this deformation tensor. So after some development, we can find this as a form of the viscosity, and we obtain what we call the Navier-Stokes equation. So the development is very simple, but it's widely available, and I don't think it will be of interest to you. But we need also to specify boundary conditions, and here we have different choice. So first, what we need to use on the wall. So there are different options. We can either use what we call no-slip boundary conditions, and this corresponds to the case where blood velocity is equal to 0 at the boundary. And this, in physics, this corresponds to the case where the adhesion force is more important than the body force or the cohesion force. So it means that the blood particles stick to the wall, and this can depend on the material of the wall. But generally, this is the law that we use, or the condition that we use. However, there are cases where we can have some slip. So the wall can be slippery, which means that the velocity at the wall can be different than 0. And this is the case where the cohesive forces in the fluid are higher than the adhesive force between the fluid and the wall. And then we need to specify some boundary conditions at the inlets and the outlets. And here we have the choice between imposing specific velocities or flow rates at the inlets. And this corresponds to the most physiologically relevant conditions that we have. So basically, we can measure the flow rates. We can measure the velocity at the inlet, which can have, for example, some parabolic form. And then we need to impose pressure somewhere, and we can impose some pressure at the outlet, but it does not change the results. And then there is two other widely used boundary conditions. So first of all, the pressure difference, where we impose a constant pressure between the inlets and the outlets, and because of this pressure difference, the blood starts flowing without having velocity imposed anywhere. And finally, we can also have cyclic or periodic boundary conditions. For example, if we want to simulate flow in a vessel, and we assume that flow keeps rolling over and over again. So this corresponds to the case where the velocity at the inlet correspond to velocity in the outlets. So I will give some examples of cases where we have used this type of models, and of course these works, they correspond to works that we did on blood coagulation, but I will not talk about the blood coagulation parts. So first we had this work where we tried to mimic some in vitro experiments corresponding to clot growth in microfluidic chambers. And in this case, we have only blood plasma. So for these experiments, we had they were done or conducted using blood plasma and not whole blood. And so we could use the Newtonian approximation here. And also we have this growing thrombus. So I will not describe how it grows, but if you have any questions, then of course it's my pleasure to answer them. But basically we have this permeable clot that starts from here and then starts growing depending on flow speed. And we wanted to study the interplay between the hemodynamics and the thrombus growth. And this is the model that we have used. And here you can see that this is the Navier-Stokes equation, but we have one additional term which describes the deceleration of blood as it crosses the clots. And we have used the constant pressure difference. So the difference between the outlet and the inlet remains constant. And also, as I said, we have used very basic Newtonian rheology model. So we assume that flow is Newtonian because here the experiments were made using blood plasma. And these simulations that I'm going to show were made using a finite volume library called InMost, which is developed by the group of Professor Yuri Vasilevsky. And basically, it allows several interesting things like the manipulation of the mesh. And this is what the simulation looks like. So these are like 2 steps before the occlusion and after the occlusion. So this layer here represents the isosurface of the thrombus that grows over time. And we can see here the velocity of blood. So here before the clot grows and occludes the vessel, we can see that there is high concentration of blood that still goes there. But here, the velocity of the clot stops the penetration of the flow, because here we are using the constant pressure difference condition. So basically, for the constant pressure difference condition, the blood can be stopped. So there is no constant flow rate, and so blood can be stopped from flowing, and the flow rate or the flow volume can decrease. So that was first example, and here I'm going to show the same problem, but when considering whole blood. So when considering non-Newtonian rheology, and also when considering localized clots. So in this case, the clots does not grow too much, does not go to the outlets, but it grows only in this particular area. And so for this particular problem, we keep everything except the rheology model. So for the rheology model, we use the non-Newtonian Caro model. So as I said, there are different constitutive models, and one of them that I personally use is the Caro model. And for the implementation, we have used this CFD library called OpenFOAM. So it's an open source library for computational fluid dynamics that is written using C++. And it's a very powerful tool. I discovered it when I was a student, and I did an internship in a consulting environment consulting firm, and since then I've been using it. It's very nice library. So here we can see the results of numerical simulations, and here we simulate the growth of the thrombus in a normal plasma and also in a COVID patient. So here what happens is when in the case of the COVID patients, the tendency of clot formation is much higher, and so the clots can grow and completely occlude the vessel. However, for the healthy plasma, then the blood flow prevents the clots from growing, and we have this sort of response, which is typically a normal response in this particular settings, because these are taken, or these are here we try to reproduce some in vitro or in vivo experiments that aim to mimic the formation of the clots. So here I want to say a quick remark regarding the effects of non-Newtonian flow. So what happens when we have non-Newtonian flow is that although the clot is localized somewhere here, but the effect of the clot can be seen also downstream. So in downstream, we still have very reduced flow, and this is observed when we consider this non-Newtonian blood rheology. And in a more recent example we are developing also with Dr. Alexey Belayev and also some other colleagues that do experiments, and I will talk about how what they do and how it's very important in terms of validation. So we are studying the movements of or the pulsatile flow in stenosid carotid artery. So we have a carotid artery, which is basically the artery that transports blood to the face and the brain and so on. So it's in the neck, and here we have one particular feature is the pulsatile flow. And so for pulsatile flow, it can be obtained using different ways that I'm going to talk about, but basically this is what it looks like. So this is what we call a typical waveform of like blood or the pressure drop, pressure difference. And in this particular case, we analyze the effect of exercise. So we have a person that is resting and a person that is exercising, and they analyze the flow of each of these person, and they obtain this they obtain this different pressure drop. And here again, we are using a non-Newtonian Caro blood flow model, so the Caro constitutive model. And we are using OpenFOAM for the CFD implementation. So this is the what the blood flow looks like for a normal person or a person that is at rest, not exercising. And we can see that this is laminar flow. And here I forgot to mention that there are some cases where flow can be turbulent if the Reynolds number is higher than 3,500. And this visualization, we can see what the flow profile looks like at this stenosid area, and also with these streamlines. So now what I want to say is how we work with these physical therapists who are conducting experiments. So they have this neck probe holder, and in this neck probe holder they insert some tool that use ultrasound to record the flow velocity inside the carotid artery. So they get some data that looks like this, and then they analyze these images to derive the Reynolds number and the endothelial shear stress. So they use ultrasound images and a neck holder, and after they collect this data, then they analyze it, but then they can derive only the endothelial shear stress, which is the shear stress at the wall, and the Reynolds number. So, but the actual movement of flow is a little bit more harder to detect, and this is what I'm going to talk about in the conclusion. But these two variables are more than sufficient to calibrate our model. So this is what we use to calibrate the model and validate it and make sure that it works, because there are some data that we use from elsewhere, like this normal waveform in the carotid artery, and so we need to make sure that we are in the good interval for the Reynolds number and the endothelial shear stress. So after this introduction, or after talking about these single phase models, the next will be to talk about the multi-phase models, which are a technique that we have started using, I think, and developing together with Professor Vitaly Volpert and also Dr. Alexey Belayev. And it's a very powerful technique, and it allows to capture some complex mechanical interactions and dynamics, like the margination of platelets. So the idea is that we use what we call the mixture theory, which was introduced, I think, in the 70s. And basically it consists in, like if you heard about the volume of fluid methods, and and things like that. So what we do is basically we consider that there are two distinct phases that are moving, or that are moving together in flow, in blood flow, which are the plasma and the red blood cells. And each of these phases has specific densities. So we have rho P and rho S, and viscosities. So we have mu P and mu S, and as well as velocities. And then we can compute the velocity using this formula, so the actual velocity. And here this phi is basically the volume fraction, which depends on the hematocrit. So it can be from 0 to 1, and it takes the value of the hematocrit. And there is one hidden equation that I will talk about in the next slide here, that allows us to keep track of what this volume fraction is doing. So basically, we apply the conservation of the momentum to these two equations. We also apply the conservation of the mass, but I didn't show the equations. But when we apply the conservation of the momentum for each of the phases, we obtain this, of course, these two equations. So here we have the Cauchy shear stress applied to the plasma, and then we have some body forces, and then we have interactions between the two phases, the plasma phase and the red blood cell phase. And we have also this term which is due to the deceleration of flow as it goes through the thrombus, so I will not talk about it. So this is for the case of blood coagulation, because this type of models is very relevant to the case of blood coagulation due to cell deposition. So as I said, there is a hidden equation here, which is this convection equation that we use to track the movement of the red blood cell phase, and then derive how many or which area is represented by red blood cells and which area is represented by the plasma. So we use this equation, and here basically it's just the red blood cells are being transported by the by the velocity of the red blood cell phase. And then, as I said, we have this source term, which consists of these three terms, if we don't consider these last ones. And first of all, we need to define the Cauchy shear stress. And here we consider two cases. So first, for the plasma, we consider that it's a Newtonian fluid. So in this case, viscosity is constant. And for the red blood cell phase, we consider that it's a non-Newtonian fluid. So as I said at the beginning, the viscosity of red blood cells and of the whole blood depends on the hematocrit. So we need to capture this. And we consider that here viscosity depends on the shear rate and on the hematocrit. So as I introduced, the asymptotic viscosities can be updated to reflect these changes. So this model was introduced by the group of Professor Antaki, and it was used to capture the machination of platelets, and then we have used it to make simulations of thrombus formation in stagnant region. And then we have also for this shear stress, we have this term which gives basically the term with the viscosity, and here I think it's the secondary viscosity. And also another term which is very important is the interaction between the two phases. And here we consider two types of interactions which are taken from empirical relations. And the first force, so this one is the drag force, which means that each of the phases can be dragged by the others. So if force is high in a phase, then it can drag the other. And we have this shear lift force, which is like the shear force when there are interactions between the two phases. So as I said, this model was introduced in this work and was used to capture the machination of platelets. And the way they did it was by considering that there is an influx of platelets from region where there is a high density of red blood cells. So when there is high density of red blood cells, then platelets get pushed away. And this is what we observe in experiments both in vitro and in vivo. So we have this margination of platelets, which makes platelets, although there are very few numbers of them, as I said, it's less than 1% for both platelets and white blood cells of blood. So these platelets, they get marginalized, they get pushed toward the walls, and they can participate in the formation of the thrombus, so they can aggregate and form the clots. And this is the model that's or the simulation that they did in this work, and they can show that they can fit the experiment for this particular case, and they can show that the platelets got so there is this margination of platelets that can be reproduced. And what is nice about this model is that it can work under different shear stress. So as I said, we have applied this model to study the kinetics of thrombus growth in aneurysms. So I will not talk about the coagulation part, but I will just talk about the results concerning blood flow and what we can learn. So first of all, we have simulated two types of aneurysms, so the bulbous and bulging saccular aneurysms. And then we have studied the effects of, in particular, the intensity of flow. So when we increase blood velocity at the inlets or the pressure drop, and when we change the geometry, and also the effects of the hematocrit, which is, I think, why this method is very powerful. It's capturing the effects of the hematocrit, which still can be done with the single phase model, but not in an accurate way like this one. So in areas where we have complex and stagnant flow and things like that, so this method becomes very powerful. And then we have validated the model using backward step experiments. And basically, this is what the simulation looks like. So we can see the formation of vortices inside of the aneurysm. And we can see that these vortices, they can rotate the red blood cells inside of aneurysms, and this affects clot growth. So the main results, or one of the main important results of this work, is that this neck size influence the rotation of blood flow inside of the aneurysm, which in turn influence the position of platelets and the subsequent clot growth. So this can give us some new insights into this complex phenomena where we have this stagnation of flow and complex rheological and geometry and things like that. So this was the one case study that we did, and more recently we have submitted a work where we study the dynamics of flow inside left atrial appendage. So left atrial appendage is a small pocket that we can find in the left atrium of the blood, and there are some cases where there are coagulation that starts in this particular area, and then it can move away and can obstruct different regions of the circulatory system. But as I said, I will not talk about the coagulation part, but what we did in this work was basically using this particular flow chart. So first we collected data from patients, and we analyzed this data. And then we have used some simulations with the validated software COMSOL multiphysics to make simulations of flow inside the left atrium appendage and also inside the left atrium. And in parallel, we have generated this left atrium with an idealized geometry, except for the appendage, which is patient-specific. And we have validated the two models. And finally, we have used this model or this multi-phase model to calculate and simulate the growth of the thrombus under different conditions. So this is a highly interdisciplinary work, and we've been working together, of course, with Professor Vitali and Dr. Alexey Belayev, but also with some other colleagues who work on this flow simulations and data processing, and also we have some discussion with medical doctors. And basically, this is what the the flow dynamics inside the left atrium appendage looks like. So we can see here that there are these vortices that form at the entrance, but there is some stagnation of flow here, so the flow does not reach these parts too much. So we tried to validate the simulations that we did with the multi-phase model against the one that our colleagues did with the capsule multiphysics. And although we couldn't reach like a very good agreement everywhere except at the entrance, but we still can see that there is, or the tendencies are the same, and this is expected because we are using an idealized left atrium, but it's still sufficient to simulate this complex clot formation. Okay, so in conclusion, I can say that these continuous models, they can offer a very strong and powerful tool to study the pathogenesis of several diseases like sickle cell, clot formation, bleeding, anemia, and aneurysms and things like that. And they can also be used to develop some medical devices, in particular stents and blood barriers and valves and things like that. So they can be used for this. And in the future, we hope that they can also be used to prepare for surgeries. And this is where I want to conclude this. In particular, I think there is a room for doing this by considering also other cutting-edge technologies, such as virtual reality and artificial intelligence. So we talk about the digital twin, which is like the surrogate model using AI, and which can allow doctors to make real-time simulations of their surgeries, for example. And this is also a technique that is used in industry and so on. But there are still some challenges which I think make the whole process of working on these projects both challenging but also fun. And first, we have always some problems with the quality of data. So as I said, some colleagues, they can provide data on endothelial shear stress or Reynolds number and things like that, but we don't know what the velocity looks like inside of the vessels, and this is why we use this type of models. And also sometimes we have data that is given as image, and we need to do some segmentation. And after segmentation, we need to process the mesh. So the mesh could or the geometry can be not very suitable for this. For example, it can have some holes. It can have like many fine meshes, and when you try to make the mesh out of this geometry, then it's not going to work. You are going to have very fine mesh, and there is also this problem with mesh, so it shouldn't be very fine, and also it shouldn't be very coarse. So these are all technical problems that we face, and also when we arrive to the stability and accuracy of numerical schemes, so how to discretize the gradient terms, the Laplacian terms, and whether to use earlier or backward schemes. So these are all questions because we can use some models or some schemes for velocity dominated flows, but they can. So we use in stability. So here I'm talking, for example, for the convection terms. And after that, we need to make sure to optimize the CPU time. So if we introduce the bad parameters, then we can run the same simulation, but instead of doing it in a few hours, then we will do it for a few days. So this depends, of course, on the mesh and also on the CFL number and things like that. And also sometimes we need to make some simplified hypotheses. So for example, when we have a moving geometry, like for example, the heart or the blood vessels, then this also whether this can have some effect on the blood flow or no. So these are also important hypotheses that we make all the time, and we need to make sure that they do not influence what refined in terms of results. And of course, in the end, what you obtain is numbers, and we need to exploit these numbers and make sense of them using post-processing tools. So these are all challenges that, as I said, also make this problem very fun to work on. And in the future, we have, as I said, this computational fluid dynamics has providing future, because I think at some point we should start using it not in research, but also in actual medical practice. So so with the help of technologies like augmented reality, virtual reality, AI, and so on, then I think in a couple of years, then probably doctors will start making surgery simulations before doing the actual things, and also start prescribing some treatments and see what it does without resorting to the in vitro assays and so on. So I think that's it. Thank you for your attention.
Yes, thank you very much, Anas, for this very interesting and quite complete overview. Like I can say, the state of the art in this really very active, very interesting, but also very complex domain. And I should say that in spite of the fact that we worked together during many years, I also learned something new from your presentation, and we can discuss a little bit now. And in particular, you mentioned that non-Newtonian fluid, considering Newtonian or non-Newtonian fluid, this can influence clot growth, if I understand correctly.
Yeah.
What you said. Could you please explain, because in fact I didn't know that. Could you please explain the difference in clot growth, how Newtonian or non-Newtonian fluid influences clot growth?
Sure, this is a very important question, and I think from just doing some simulations, I have noticed that when we use non-Newtonian flow, then there is this part where, despite the fact that there is no clot here, but the flow still, or the velocity is still very reduced here. And I also seen it in other works, for example, with stenosis and things like that. And I think this is particularly important for the case of when we have constant pressure drop, because having this large area where blood velocity is reduced might accelerate the development of the clot, because we know that when and I think this is the work that I've been doing under your supervision in the PhD. So when the clot is large, then also it grows quickly, because it reduces the velocity of the blood. So yes, I think this is very interesting area of research, and I think that probably there are still some interesting insights to and probably some other effects, not only this one, but also some other effects, especially when we consider complex geometries that could be interesting to explore.
So can we say, according to your explanation, can we expect that clot growth will be more important in the case of non-Newtonian fluid?
I think so, at least in the case where pressure drop is maintained constant, because in this case, as I said, this area where blood velocity will be very small will be larger, and this, I think, will promote clot growth. Yes.
It can be also related to the flow velocity and to the vessel size, because usually what we understand that in large vessels, non-Newtonian properties are less important, and rather often blood is considered as a Newtonian fluid. Is it? What do you think? Is it correct? As this?
Absolutely, I think especially in the case, because for large vessels also the velocity of flow increases, and probably the effect of non-Newtonian fluid could be less important in this case.
Yeah.
So,
by the way, sorry to interrupt you. Please continue. I just had an idea. I'll continue later. Please go ahead.
I think absolutely. I think having like basic simulations where we change the shear rates and consider like Newtonian versus non-Newtonian blood flow rheology might lead us to some interesting conclusions, I guess.
Yeah, but what I thought during your answer, it's about our ongoing work, which you do together with Alexei Belayev in particular, about also platelet activation. How non-Newtonian, is it possible that non-Newtonian properties of the fluid can influence platelet activation in a different way compared to Newtonian fluid?
Yeah, I haven't thought about this point. I think probably
maybe Alexei,
yeah.
If Alexei is still here, because it's also related to von Willebrand factor. Alexei, if you listening to us, could you maybe comment on that?
Yes, hello to everyone, and thank you, Anas, for this comprehensive lecture. About this question, I've been thinking about it for a while, whether the collisions between red blood cells can enhance platelet activation through this mechanical way. Well, perhaps it may act nearby the vessel walls in case when we have high hematocrit. But well, generally, it is believed that increased hematocrit leads to the greater accumulation rate for platelets to adhere and assemble near the damaged endothelium. Regarding the von Willebrand factor, I have seen several ideas, maybe that enhanced shear diffusion is observed in cases of high hematocrit, but generally I haven't seen any thoughts that rheology can be in particular important for von Willebrand factor activation, because, well, partly because there is a depleted layer near the walls usually formed due to lift forces that Anas talked about. And this depleted layer is believed to have rheology similar to clean blood plasma rheology, which is almost Newtonian. However, in complex geometries, it may be quite relevant that platelets can collide with red blood cells, and also red blood cells can collide with already formed platelet aggregates, deforming it and leading to some not very well studied effects.
Yes, thank you, Alexei, for this comment.
If I may, I wanted to ask.
Yeah, sure. Yes, please, of course.
About this particular slide, I wonder what was the difference between COVID-19 patients and healthy patients in this model.
Yeah, so as you know, we have this coagulation model, which basically describe what's happening in terms of the biochemical reactions that culminate into the production of the fibrin. So what happened is that we have this experiments from a mice where they injected some some autoantibodies from a COVID patient. And these autoantibodies, they act on the coagulation cascade, so they activate the neutrophils. And these neutrophils, they start the coagulation from the contact pathway. And so basically the reaction for the production of thrombin is accelerated when we have this COVID patient. So we simulate this using this thrombin generation model and fit into the thrombin generation curves. And then we took these parameters, and basically here we just did two simulations with the parameters, the standard parameters for a normal patient and the modified parameters, coagulation parameters for the COVID patients.
Yeah, I see. So basically the mechanism involves cellular component of blood, namely white blood cells, right? But you consider only the thrombin, only the result of this involvement of neutrophils, right?
Yes, so the model that we use, and I think you are familiar with it, has only 3 equations. So there is prothrombin, factor 10, and thrombin. But the parameters that we use in this model can be fitted to reproduce any type of thrombin generation curves. And so we can capture the effects of several things like these autoantibodies by modifying these parameters. And so we did these modifications and we did validation of this thrombin generation curves. And this gives us a way to implicitly capture the effects of the COVID-19 autoantibodies.
Yeah, I see. But do you think that introducing neutrophils directly in the model will give some change or some relevant difference?
Yeah, that's a good question. I haven't thought about it, to be honest, but I think in terms of neutrophils, I think they can be introduced like another protein or like the platelets. So I don't think in terms of mechanics they have anything that makes them different in terms of diffusion and convection. So I think it will be like adding one additional equation, probably with a different diffusion coefficient, and I don't think it will influence the results too much. So yeah, I think probably I don't expect to see like major changes related to spatial effects because yeah.
Well, if I can just comment also on this question, as far as I remember, for these COVID-19 patients in particular, blood coagulation is reinforced due to neutrophil traps.
Yeah.
Anas, do you remember that? So it's related to neutrophils. Probably it plays important role. I'm not sure how to take it into account in the model. It can be, as Anas explained, it can be taken into account in the model like efficiently through parameters, but how to introduce it more explicitly, I don't know. Maybe it is possible.
I see. Thank you.
Well, we have really many questions to discuss. I'm not sure that we can ask and discuss all of these questions, but maybe I can ask at least one of them, and it is also, or maybe a couple, maybe 2 of them. All right. And the first one is also related to this ongoing works with blood oscillations, pulsations, which you mentioned. I understand that this work is not finished, but what do you expect? Can blood pulsation influence clot growth? So can it be the difference, in fact, in clot growth due to blood pulsations?
Yeah, this is a very good question, and I think we discussed it also with Alexei in the correspondence through emails that we did. So it appears that this activation of von Willebrand factor that is attached to the wall can be different when we have antegrade and retrograde flow, which means that when we have this antegrade flow, then of course the von Willebrand factor will be elongated, and when we have retrograde flow, then in this case probably it will be coiled. So in this particular case, I think with when we have this pulsation, then it might cause some retrograde flow, which I think can influence the general results. And to be honest, I don't know to which extent and whether this effect will be important, but I think it's a very interesting feature to have in the model and also to study, because also in terms of methods development, I think also working with Alexei and the simulation that he's doing with his model, I think this gives us very high fidelity description of all these processes.
Yes, can we expect that if you take into account these pulsations, and von Willebrand factor, Alexei, maybe it's also the question to you. Then due to this, so due to pulsations, there is high flow, high velocity, like phase and low velocity phase. During high velocity phase,
Yeah.
so von Willebrand factor is strongly activated, and at this half period, it is possible due to this activation that more platelets are attached compared to the average flow without oscillations. So maybe you can comment on that, or maybe Alexei can comment on that.
Yes.
So my sorry, to make it more clear, my hypothesis is that oscillations, blood pulsations, at least in some cases, can reinforce clot growth.
That's interesting question. I think, in my opinion, and maybe Alexei can comment after. So I think the more the more logical, at least to me, it seems that the pulsations probably will reduce coagulation, because when we have
excellent. We have opposite hypothesis. So it's really an interesting point to check.
I would like to add that it's not very obvious what happens to platelets in pulsatile flow, because the GP1B receptor-related aggregation is usually thought to be reversible, and when the shear stress drops down to low values, the von Willebrand factor gets deactivated, and platelets normally tend to deaggregate. And what happens if we have such a pulsatile flow in complex geometry, especially when we have the flow reversal effect near the wall, antegrade and retrograde flows? So it's not very obvious whether it will enhance or inhibit thrombosis, but that's why we're doing modeling. I think it has not been done yet numerically, and the number of experimental papers is quite limited. So yeah, it's very interesting thing to study.
So if I understand correctly, what you say, Alexei, is that during this high flow, high velocity flow, well, this time period, von Willebrand factor is activated, platelets are attached, but then the next half period, they will be detached. Is it what you say?
Probably yes, because there is an experimental study of Fu and colleagues who studied using microscopy. They studied the elongation and activation of individual von Willebrand factor multimers, and they have shown that attachment of GP1B receptor happens during the high shear stress phases, but when the shear stress drops to zero, this protein quickly detached from the von Willebrand factor, and this is reversible. So perhaps,
yeah, but this is reversible. But which of them is irreversible?
Perhaps platelet activation is somehow irreversible. Maybe extremely high shear stress will trigger platelets to activate the integrins in irreversible manner. So this may be the part, non-linear part, which leads to the overall thrombus development. But I'm not sure about that. It's just a hypothesis.
Yeah, very interesting question, very, very complex. Well, we don't have very much time, so I will ask probably among all questions I have, probably the last one. It's about the experiments. Anas, you mentioned that, as far as I understand, these are maybe your colleagues at the university or maybe some other, but somehow related to your team, about these experiments where this Reynolds number and shear stress is measured in the carotid. Could you maybe return and shortly explain how they measure? So, because, well, I thought very little I know about that. I thought that such kind of experiments, not only experiments, but also in clinics, they use that kind of a Doppler effect to measure flow velocity in the carotid artery. Is it something similar or it is different?
Yes, I think they use this Doppler images and ultrasound. So I think it's the same, but they have some sorts of approximations that they made, some statistical and numerical methods that they use. I think using this Wormsley numbers and things like that. So they can derive this data out of these complex figures that we can see here. So so yes, I think it's Doppler and ultrasound images. But here what they do is these people, they make the exercise. So they do like they run or they do aerobic exercise, and they have their they put this on their neck using a holder, and they can record these changes in real time. So this is the research that they do. And as I said, they can only measure some few characteristics like shear stress, Reynolds number, and things like that. But in terms of research, this also leads me to like previous question regarding pulsation and turbulence flow. So they had some research showing that retrograde flow associated with pulsations can be beneficial to endothelial dysfunction. So they work on this endothelial dysfunction, which can lead to problems like atherosclerosis and things like that. And the popular or the main belief is that retrograde flow is harmful and promotes endothelial dysfunction, but they show that it's beneficial. It actually can prevent it, and there are some conflicting mechanisms depending on the intensity and things like that. So I think this is what I can say. But yeah.
Well, again, very interesting question. What I thought discussing with you and this related to this ongoing work, that the conclusion will be that physical exercises are bad for our health. but maybe my conclusion is not exact. I'm not sure. because it can promote physical exercises, can promote blood coagulation and clot formation. Is that it correct?
Yeah, I think that's one of the expected conclusions, but I think it will depend on many things, like this particular person and how, like the blood pressure and comorbidities and things like that. But generally speaking, I think it's high intensity exercise, not like moderate exercise. So it's like people who do weightlifting and cardio and things like that. So this is like the type of exercise that can be harmful, but again, it's only for shear induced platelet aggregation. It can be beneficial for, I think, circulation and endothelial function and coagulation in other areas, but with shear induced platelet aggregation, it can be harmful, probably in some cases.
Yeah. So we should be cautious about that. Yes, please. Yes, sorry.
There is a joke that overall, after the hospital, people die more frequently than without a hospital. So doctors are dangerous for people because people die after meeting doctors.
Yes, good remark, Alexei. Okay, please, other questions or comments from our listeners?
Anna, I wanted to ask you about the force which you imply in the two-phase model. Could you please, there's certain forces, as far as I remember. Could you please show the slide and with a force acting on the on the red blood cells from the plasma.
Can you repeat which slide? I'm having some
when you have two-phase model.
Yes.
Equations for
two
two-phase
phases.
for the two-phase model.
This one? Or
No,
Safman force.
no, it's not what. No, it's not what I see. Yes, this one. Yes, this one.
And next slide, please. Next. Next slide.
Anas,
Next slide, yes.
next. Show the next. No.
No.
Because there's there is time delay in the communication.
Yeah,
I think it's 28. Slide number 28 with the lift force.
28, yes. Yes.
It's very interesting. It's an example of instability due to time delay.
Yeah. Yeah. Here. Well, as far as I remember, Safman force. Well, could you please comment this? Because as far as I remember, Safman force is something like a ball is the force which act on the droplet or particle which is alone in the channel, and its relation for this force is derived for the soul, for the one particle which interacts with the flow and with walls. And here you imply this force for crowded suspension.
Yeah.
Could you please comment on this?
Yeah, I'm sorry. I have some problems. I haven't heard very well, but if I understood the question exactly, is how this can translate to like how the flow acts on particles, and also how particles interact with the wall. So
sorry, Anas.
yeah,
Anas. Sorry, I will repeat the question as I understand it. Alexei asks about Safman force. That according to him, Safman force is a force acting on one particle. But you apply this force to the continuum medium, to the whole phase of particles. So could you comment on that?
Yeah, that's a very good question. I think it's also the same for the drag force, and this is also how we apply drag force on a single particle. And I think this can be obtained using this generalization. For example, if we take a flow particle that moves across a stream, and we try to move from the Lagrangian to the Eulerian approach, and using this by considering this derivative, like the material derivative, and then moving to the normal derivative. So this is exactly how we obtain the Navier-Stokes equations, and also the conservation of the mass and momentum, and things like that. So yeah, I think it's I think the same formula, and basically this type of, for example, if we look at this force, it's the difference between a particle belonging to the velocity of a particle belonging to the fluid, to the plasma phase, and the other belonging to the red blood cell phase. And this difference is similar to what you said in terms of the force of fluid acting on a particle. So it's I think the same approach that we use here.
And did you compare your concentration gradient of erythrocytes across the flow with experiment in this model?
Compare the gradients of the force. Is that what you said? Sorry.
No, concentration.
As far as I understand, the meaning of all this activity is to describe that the concentration of erythrocytes differs across the flow. Yeah.
Yes.
So so did you obtain this profiles of erythrocyte concentration, and did you compare them to the experimental data?
So you are talking about the concentration of platelets, right?
No.
Or
No.
Erythrocytes.
erythrocytes.
Red
Erythrocytes. Okay. Yeah, that's a very good question, and I don't think we have this level of experiments, but we still did. And I remember when we were working on this, we have considered many experiments and also some other computational works that use particle methods like dissipative particle dynamics, and we could see the same dynamics that is observed with a few differences. So, for example, we can see this type of circular shape of the red blood cells that was also obtained, I think, in another work by I think a team from the Netherlands. So yeah, I think we have worked on this type of validation, although it's very difficult to find like experimental data that can show you where red blood cells are located. So most of the validation that we did was using other modeling works that use some fine-grained methods like dissipative particle dynamics.
But I would like also to add that yourself you did comparison with the experiments on backward step flow. Do you remember that? It was exactly in this work where for this aneurysm modeling,
yeah,
as for the validation, you did this backward step model, and there are experimental data on the distribution of red blood cells.
Yes, I think for this work we didn't have data on the concentration of red blood cells, but we have used it for the validation of the clot growth dynamics, which in turn depends on the platelets margination, which depends on the distribution of red blood cells. So it's not like direct validation, but I think it's all of these observed dynamics can be related to where red blood cells are located and where platelets are located.
Okay, thank you. Thank you, Hans.
Well, if there are no other questions, then I would like to thank Anas again for this very interesting presentation and for the interesting discussion. And I hope that Anas, you will keep us updated about your work and our future seminars. So thank you. Just a moment. Something in the chat. What is that? Just a moment. Okay. Our listeners thank you, Anas, for the presentation. So thanks to everybody and have a good day. Bye now.
Thank you for having me. Bye.
Bye.
Thank you. Goodbye.
Thank you very much.
Thank you.
Bye.
Bye.